I am very glad to have this opportunity to present the label-free immunoassays that we established in our clinical lab for SARS-CoV-2 serology tests. The content of this webinar will cover an introduction to the label-free immunoassays, and then we'll talk about the SARS-CoV-2 serology tests, including the methodologies and the test results. And then we will have some discussion points to share. In order to understand label-free immunoassays, I think we first should take a look at the label-free technologies on which the label-free immunoassays are established. Label-free here is used in contrast to the conventional immunoassays in which labeling is required. So let's take a look at a conventional immunoassay. Here the example is ELISA. And as we know, ELISA requires a reporter molecule attached to the immune complex that is formed in the sample well. And the reporter molecule could be an enzyme, for example, HRP or ALP. And the enzyme will catalyze some reaction to generate a color in the sample well, and you can measure the color intensity to quantify the analyte in the sample well. And not only for ELISA, in other conventional immunoassays such as fluorescence or chemiluminescence immunoassays, a reporter molecule is required. However, if you use label-free technologies to run immunoassays, then you don't need a reporter molecule. The most classic label-free technology is called surface plasmon resonance, or SPR. And SPR senses the refractive index change at a metal solution interface. So if you have an antibody and an antigen, you want them to interact at a metal solution interface, then you can use SPR to monitor the entire time course of this immune reaction in real time. And you don't need to attach any reporter molecule to the immune complex in order to see the signal. On the contrary, you actually are able to observe a signal from time zero and see the full time course of this immune reaction. So this is very powerful. However, SPR requires a complicated fluidic system, so it is not widely used in the situations of running immunoassays. It's more used in characterizing biomolecular interactions. After the year 2000, a newer label-free technology called Biolayer interferometry, or BLI, came to the market. And this is very powerful and convenient. BLI uses a sensing probe, and the immune reaction can happen at the surface, at the tip surface of the sensing probe. So as you can see in this graph, the incident light and the reflected light travel in the sensing probe, and they can generate interference pattern. And through the pattern, we can characterize how many molecules bound to the tip surface of the sensing probe. The BLI technology senses the optical thickness change at a glass solution interface. This is the way we quantify the analyte in a sample well. On the right graph, as you can see, the sensing probes can be lined up in an array and dip into sample wells to measure the quantity of analytes in the sample wells. So this is a very good format for clinical labs use. So that's why the new types of label-free technology could be a powerful tool in clinical diagnostics. And here it is an illustration of how we apply the dip in and read type of label-free technology to establish label-free immunoassays in clinical labs. So the technology we use is next generation biolayer interferometry. In other words, it's thin film interferometry. So we use a quartz glass sensing probe as shown in this graph, and we actually carry out immune reactions on the sensing probe surface. And when molecules bind to the surface, the interference pattern changes, and the signal is obtained from the interferometry. And on the right, it shows how a typical label-free immunoassay can be carried out. Let's say if we have a sensing probe which already contains capture molecules on the surface, and when we bind a layer of antigen molecules to the capture molecules, we will see a phase of signal increase. And then if we bind another layer of antibody molecules to the surface, actually to the antigen molecules on the surface, we will see another phase of signal increase. So this is very powerful because we can see the entire time course of the immune reactions, and we know exactly what is happening in the entire time course. And by seeing this full time course in real time, we have the advantages of these label-free immunoassays. First, it's easy to do assay development and troubleshooting. And second, it allows for rapid quantitation based on the initial binding rate. And this is a very important feature that we applied in some antibody drug monitoring assays as reported in this paper. So today, since for the SARS-CoV-2 assays, we are not using this feature, so I'm not gonna explain it in details, but I just want to mention this is a very important feature for label-free immunoassays if you want to achieve rapid analysis of your samples. Okay, after the brief introduction of the label-free technology and label-free immunoassays, I think now you understand that we are able to monitor immune reactions in real time, and we can see the full time course of the particular immune reactions. So this is the feature we are gonna use in the SARS-CoV-2 serology label-free assays. And now let's move into the second part. Let's talk about the SARS-CoV-2 serology. First, I want to talk about the study of IgG and IgM concentrations in our lab. This study was not carried out using label-free immunoassays, but it laid important foundation to our next steps using label-free immunoassays. And this work was published a few months ago in Clinical Infectious Diseases. The graph on the left shows that in multiple COVID-19 patients, the IgM concentrations labeled in black dots typically increased in the first couple of weeks after disease onset, and then started to decline, while the IgG concentrations increased a little more slowly than the IgM concentrations, but then plateaued at higher levels. So this observation is consistent with our knowledge of infectious diseases, in which IgM typically is the first isotype of immunoglobulins to appear, and then IgG becomes the most important isotype. And the graph on the right shows that IgG concentration could be used as a differentiator for severe and non-severe cases, as the ICU patients typically showed higher IgG concentrations than non-ICU patients after 2 weeks. With the knowledge of the IgG and IgM concentrations, we want to move a step forward to study other serology parameters, for example, the antibody avidity and antibody neutralization activity. This slide illustrates the label-free immunoassay we established to measure IgG avidity. By definition, avidity means how strong an antibody binds to its antigen. And in clinical diagnostics, we typically measure antibody avidity in a simple way. We first form the immune complex of the antibody and antigen, and then we apply some harsh conditions to dissociate the immune complex, and we measure the percentage of the immune complex remained in the end, and this percentage is called avidity index. So this process is illustrated here in this graph. So in the label-free immunoassay, we have a sensing probe coated with the antigen, which is the receptor binding domain of the SARS-CoV-2 spike protein. And we place this sensing probe in patient serum. And the antibodies in the serum will bind to the surface. And these antibodies are specific against the SARS-CoV-2 spike protein receptor binding domain. And not only IgG, also other isotypes like IgM will bind to the surface. And we observe a signal increase in this step. And then we place the sensing probe in either running buffer or the dissociation agent, like urea. And we will see a signal decrease. That means the dissociation of the immune complex. And finally, we place the sensing probe in an anti-human IgG solution. And the anti-human IgG will bind to the IgG molecules on the probe surface and make signal increase. And the final signal increment is our readout, which is correlated with number of IgG molecules on the surface. To calculate the avidity index, we just need to calculate the ratio of the readout using a dissociation reagent versus the readout with just running buffer. And this gives us the percentage of the immune complex remain on the probe surface. And some real data are shown on the right. As we see, 8 samples are measured in parallel, and in the dissociation phase, with running buffer, you see very minimal signal decrease. But when we apply the dissociation agent, urea, you see significant signal decrease, which means the dissociation of the immune complex on the sensing probe surface. The results of the label-free IgG avidity assay are shown in this slide. And the data were recently published in the Journal of Clinical Infectious Diseases. We measured 168 serum samples, and in graph A, we plotted the data against the days after disease onset. And as we can see here, the overall trend is that the IgG avidity increased over time and then stayed at a higher level. And the time window we measured is about 3 months, 90 days. And in figure B, more specifically, we see the timed sample series of 13 individual patients. And in each individual patient, we see the trend of IgG avidity increase, and then it stays at a higher plateau level. In graph C, we see there might be a significant difference on IgG avidity between ICU and non-ICU patients. So the overall conclusion is that we know IgG avidity increases over time and then stays at a higher level. And we're talking about the time window about 3 months, which is particularly meaningful because in early COVID-19 publications, usually people measure the serology within 1 or 2 months. And now we extended to a longer time, and it helps to monitor the serology parameters over a longer period of time. So far, I have introduced how we measured the antibody concentrations and avidity in patient serum. As we know, both concentration and avidity are components of the overall antibody neutralization activity, which can be quantified by the neutralizing antibody titer. And the next question is, how do we measure the neutralizing antibody titer in each serum sample? We followed the concept of surrogate virus neutralization tests to design a label-free immunoassay for this purpose. The label-free immunoassay is illustrated in these graphs. So in each measurement cycle, there were 2 steps. And the first step is shown on the left, and the second step is on the right. So let's take a look at the first step. We actually use the same sensing probe coated with the receptor binding domain of the spike protein, and we place the sensing probe in a serum sample, and all the antibodies against the receptor binding domain of the spike protein will bind to the sensing probe surface, making a signal increase in this phase. And then we place the sensing probe into a solution of angiotensin converting enzyme 2, the ACE2. And as we know, ACE2 is the binding target of RBD. So if the RBD on the sensing probe surface is completely neutralized by the antibodies, no ACE2 will be able to bind to the sensing probe surface. However, if the RBD is not completely neutralized, then some ACE2 will bind to the probe surface. So the signal increase in this step is actually our readout, which is correlated to the amount of ACE2 bound to the surface. In other words, it's the amount of RBD that is not neutralized by the antibodies. Then let's take a look at the step 2 in the same measurement cycle. In this step, we measure the full capacity of the sensing probe to bind ACE2. So we actually use exactly the same sensing probe and do a regeneration process to remove all the antibodies bound to the RBD. And then we place this regenerated sensing probe in the ACE2 solution to measure how much ACE2 can fully bind to the sensing probe surface with RBD. And the signal increase is our readout. So by calculating the ratio of these two readouts, we actually get a percentage, which we call neutralization index. So this percentage actually means how much RBD was not neutralized by the antibodies in the first step. So this is the process of obtaining the neutralizing index. And for each serum sample, we actually measured neutralization index at multiple dilution ratios. This slide shows the data of 8 samples measured in parallel. The samples were diluted in the running buffer at 6 different dilution ratios, and the neutralization index at each dilution ratio was measured. So there were 6 measurement cycles, and I'm just showing the first steps in these cycles, and only one second step as an example, because the second step of all the measurement cycles look similar. And in the first steps, as we can see here, the last phase of ACE2 loading increased as the dilution ratio increased. That definitely makes sense, because the higher dilution ratio resulted in the less neutralization of the RBD on the sensing probe surface, so that more ACE2 could bind to the sensing probe surface at higher dilution ratio of the serum sample. After measuring all the dilution ratios of each serum sample, we could plot the neutralization index against the sample dilution ratio as shown in the examples in the slide. There are multiple samples in each graph, and it's easy for us to quantify in this way the neutralizing antibody titer, the IC50, by measuring the dilution ratio at 50% antibody neutralization index. And this neutralizing antibody titer represents the overall antibody neutralization activity in each serum sample. This is an important slide as it summarizes what we have measured so far on neutralizing antibody titer, IgG concentration, and IgG avidity. The first row of graphs show the results of 268 serum samples, and the results are plotted against the days after disease onset. So we have extended the time window to over 200 days to obtain a more comprehensive understanding of the trend of these serology parameters over time. As we see here, the neutralizing antibody titer and IgG concentration, it seems they move in the same trend just by looking at the overall results of all the samples. It seems they go first went up and then went down. For IgG avidity over time, it seems the observation is consistent with what we showed earlier. that the IgG avidity just increased to a higher level and stayed there. And in the second row of the graphs, we actually picked the sample series of 26 individual patients and show the trend of these serology parameters here. And again, it confirms our observations from the first row of graphs. The neutralizing antibody titer and IgG concentration of these sample series, they both quickly reach a high level, and then there's a trend of declining. And for IgG avidity in these sample series, it just reach a high level and tend to stay at that high level. To better illustrate this observation, we picked 15 patients with a relatively large number of samples and plot the results of these sample series in individual graphs. As shown here, the IgG avidity in most cases tends to reach a high level and then stay at the high level, while the IgG concentration and the neutralizing antibody titer tend to move in the same trend. They first go up to reach a high level and then start to decline. As we know, the antibody concentration and antibody avidity both contribute to the overall antibody neutralization activity. So the conclusion could be that the IgG concentration is the deciding factor to impact the antibody neutralization activity in a serum sample. So these are pretty much all the data I want to present today. And based on these results, we have a few discussion points to share. First, we demonstrated that the label-free immunoassays based on the TFI technology worked for clinical testing of antibody avidity and antibody neutralization activity. And we observed SARS-CoV-2 IgG avidity increase over time. And given that the IgG avidity was strong in ICU patients after 1 month, the antibody-mediated disease enhancement, usually triggered by suboptimal antibodies, may not play a role in COVID-19 disease progression and severity. And we also observed the SARS-CoV-2 IgG concentration increased first and then decreased in 1 to 2 months. And similarly, we observed the serum neutralizing antibody titer increased first and then decreased in 1 to 2 months, which was consistent with the IgG concentration. Given that the antibody neutralization activity depends on both antibody concentration and avidity, the decline of antibody neutralization activity could be due to the decline of the antibody concentration, while antibody avidity stayed strong over time. With that, I would like to acknowledge my colleagues at UCSF, San Francisco General Hospital, Dr. Carol Lynch, Dr. Alan Wu, Cassandra, and our clinical lab team. I also want to thank my collaborators in Gator Bio, Hong, Bob, Catherine, Anthony, Paul, Mitch, Jesse. You guys are great people to work with. And thank you all for attending this webinar.
Hey, since we still have some time left, I welcome questions.
Yes, it looks like we have a few submitted. So if anybody has any questions, feel free to type them in, but we can go to the first one now. So the question is, can you elaborate on the principle of using urea and running buffer ratio to evaluate avidity?
Okay, so yeah, let me explain a little more details of how to run the avidity assay. Actually, we borrowed the idea from some existing clinical lab tests. For example, like toxoplasma tests, in which the tests actually follow the same idea to measure the avidity index to judge if the infection happened recently or happened a longer time ago. So what we do here is we first, as I described in the idea, we want to form an immune complex in our case on the sensing probe. So as you can see here, we have immune complex formed in this step. And then we either place the sensing probe into running buffer, or we place the sensing probe into urea. So these are actually two different processes. in actual experiment, we actually first place it into running buffer, and then we complete the process. And then we regenerate the sensing probe to remove all the IgGs and anti-IgGs bound to the surface. So we just restore the state of this sensing probe, and then we place into the same specimen again, and then we put it in the urea. So that these are actually two different, steps in this one measurement cycle, and then we calculate the ratio in these two steps. So basically, I want to mention here is we have a very good regeneration process to restore the state of the sensing probe. That's necessary. Otherwise, if the sensing probe is changed between these two steps, then we cannot do this ratio measurement. Thanks to label-free technology, we actually monitor the entire time course. So we actually compare the IgG loading level over different steps using the same sensing probe, so that we can easily tell if the regeneration of the sensing probe worked well or not. I hope this answered the question.
We have another question coming in. Can you explain how you control for the competitive binding of IgM for your label-free IgG avidity assay? So for clarification, the competitive binding of IgM to the RBD.
That's a very good question. Actually, we didn't, and we couldn't. So once we place the RBD probe in the specimen, we know that IgG, IgM, or even other antibody isotypes like IgA will all bind to the surface if they are against RBD. But the good thing is we have another step. In this step, we use a specific anti-human IgG to bind to the IgGs on the surface. So IgM and IgAs will not be detected in this step. That's why we use the readout from the last step, not the readout from here, because we know the readout from IgG loading actually includes other isotypes. So that's why we just stick to the readout in the anti-IgG measurement or the calculation of the avidity index.
We have another one. For the development of the avidity assay, I would imagine that you would need to standardize the loading and association-dissociation times early on and keep them the same for all indices being compared. What considerations are most important when you are thinking about good assay design for response and time in this assay?
Yeah, so again, for both assays, we measure, we actually have multiple steps on one sensing probe to measure one specimen. And especially in the neutralization assay, we actually even have multiple dilutions of one specimen. And we just use the same sensing probe to measure that. So I think the number one thing technically is we need to make sure the regeneration really works well. And the good thing is we despite the assay development that we did, just using different conditions, in the real measurement, because we are able to see the entire time course. So we actually compare, as I mentioned, in the avidity assay, we compare the IgG the antibody loading levels, in multiple steps when we measure the same specimen. And also in the neutralization assay, we were able to measure it even in a better way, because we in each measurement cycle, we not only have antibody loading curve or association curve, we actually also have a second step of the surface capacity test using just the bare RBD probe against ACE2. So in this case, we just have like two pure agents to react. So it's very easy to tell if the sensing probe still performs to the same quality level, because we can tell how much ACE2 loaded to the surface. And typically we see very good reproducibility of a same sensing probe over multiple regeneration cycles. Usually we have CV within 10%. Sometimes, we have most of the sensing probes with the ACE2 loading CV within 5%. So that's actually very good data and showing just the reproducibility of the sensing probe. I think once we confirm the reproducibility of the sensing probe, I think that's actually solved most of the concern, because the rest of the part is just, we want to carry out the immune reactions with minimized non-specific binding. And we actually use a running buffer with a pretty high concentration of PSA to minimize that. And we also dilute the samples in the running buffer just to minimize non-specific binding. I think these are the concerns that we took to optimize the assays.
Perfect. We're getting a lot of questions. I'm not sure if we'll be able to get to all of them. Let's go to the next one though. Can you please comment on the chemistry of the RBD to the biosensors? Talk a little bit about what they are. I
see. That's a very good question. I'm not sure how much Gator wants me to release about this information, because I know you guys may convert it to a product later. I think I think what I can tell is basically the idea is we obtained RBD from a large vendor which produces recombinant proteins. And then the RBD, we just labeled it with some molecules, for example, biotin. And then we can use biotin streptavidin interaction to immobilize it. But I know Gator also has other ways to immobilize molecules. Actually, you have a number of choices. So I think in the future, if there's even a more convenient or more efficient way of immobilizing RBD, then I'm sure Gator will take that to do it.
Another question: how do you explain the increase in avidity over time? IgG maturation? Is there any way to tell early on to predict ICU necessity rather than after the fact?
That's a very good question. We actually tried. We are currently working with the UCSF and other departments in UCSF, and we are participating in a nationwide clinical trial called CAPRI to trying to get more information about avidity versus the severity in clinical cases and the time. So far, this is the best conclusion that we could find. Like within 1 month, to be frank, it's really there is some difference, as you can see in this graph. But for clinical diagnostics, it's hard to find a cutoff just to tell if this ICU patients versus non-ICU patients. Over 30 days, it's better, but there's still some error, as you can see, because there's some overlap. So our idea is we want to collect multiple parameters of serology, not only the avidity, but also the surrogate virus neutralization titer that we, in this graph, I'm sorry, actually here. Yeah, so we're just trying to use multiple parameters, see if we can get like an algorithm using them to help clinicians to judge the potential disease progression over time at an early date. So that's actually in process. We are collecting more data, hopefully, to get to that target.
Perfect. Okay, I think we'll do one last question. Is there consideration of the binding kinetics of the neutralizing antibody versus the binding kinetics of the ACE2 as a reference to obtain your index calculation?
I'm not sure if I fully understand this question. I think this question is about the neutralization test. Catherine, do you mind re-elaborate the question?
Let's see. So is there consideration of the binding kinetics of the neutralizing antibody versus the binding kinetics of the ACE2 as a reference to obtain your index calculation?
Oh, I see that. Yeah, this is a great question. Yeah, I think whoever asked this question might have a background in the label-free biomolecular interaction measurement. Actually, we did consider that if we can extract information from the binding kinetics. Yeah, for those who ever did label-free characterization of biomolecular interactions, that we actually can get K on, K off, and the equilibrium constant KD just from the association and the dissociation curves. However, in this case, we tried, and apparently, in this step, there's no accurate kinetic measurement could be done because first, there's a huge number of antibody molecules in many patient specimens. So the mass transport effect is one concern. And second, there are multiple analytes. As I said, all the antibodies against the RBD will bind to the surface. So it's a mixture of all the kinetic information, and we could not extract kinetic constants from in this step. And in ACE2 binding, if we control the conditions, we might be able to get some kinetics because ACE2 is a pure analyte here. So we actually thought about that, but since we were able to get a readout and follow our diagnostic tradition to get a neutralization index, so we haven't done much, haven't gone so far just in trying to measure the kinetics between the ACE2 and the RBD. But I think there have been some reports using label-free technology. I mean, previous publications, they characterized the binding kinetics. Yeah, actually, I used that to decide the ACE2 concentration that we use. But I understand this question is not only just between the pure RBD and ACE2. After the binding with loading of the antibodies, can we extract information from the binding kinetics? I think there might be a possibility of doing that if the antibody can play a role in this kind of binding, for example, like a competitive inhibitor or non-competitive inhibitor. So that's a very good idea. We will, in the next step, if we have the bandwidth, we will try this idea. But so far, we only just stay with the amount, the increment of signal as a readout to calculate the neutralization index.
All right, well, I want to thank you, Dr. Luo, for giving this fantastic webinar. It generated a lot of conversation, a lot of questions. And I want to thank everyone for attending. And like I said at the beginning, this is being recorded, so we will have a link to it and it will be emailed to all of you. So thank you again.